Welcome back to my channel. This is episode 6 of the Break It Down series, Nucleic Acids and Protein Synthesis. Let's then dive straight into the video.
But wait, let us all take a moment to like, comment, subscribe if you haven't already. And now let's watch and learn. Question 1. Which row represents the correct features of the nitrogenous base cytosine? Over here, remember Gapur, which means that only guanine and adenine are purines, the rest are all pyrimidines. So since cytosine doesn't belong in Gapur, because Gapur stands for guanine and adenine are purines, therefore cytosine must be a pyrimidine. Hence, under the column is a purine, it should be false, leaving us with the options being either B or D. And a key thing you must know is that all purines have a double ring structure, whereas pyrimidines have a single ring. Therefore, since cytosine is not a purine, it will not have a double ring, but it will have a single ring. Therefore, under the column has a single ring structure, it should be true, leaving us with the answer being B. Has a single ring structure true, is a purine false, joins its complementary base by 3 hydrogen bonds is true, because C, which is cytosine, binds with guanine using a triple bond, that is 3 hydrogen bonds, whereas A binds with T or A binds with U using only double bond. Therefore, the answer is B. Question 2. Bacteria were grown in a medium containing 15 nitrogen. After several generations, all of the DNA contained 15N. Some of these bacteria were transferred to a medium containing the common isotope of nitrogen 14N. The bacteria were allowed to divide once. The DNA of some of these bacteria was extracted and analyzed. This DNA was all hybrid DNA containing equal amounts of 14N and 15N. In another experiment, some bacteria from the medium with 15N were transferred into a medium of 14N. The bacteria were allowed to divide 3 times. The DNA of some of these bacteria was extracted and analyzed. What is the composition of this DNA? Although a long question, you have to break it down into its key parts. So I have underlined over here. First, it was grown in a medium containing 15N, which is the heavy nitrogen. And after it was transferred to the medium containing light nitrogen, that is 14N, and allowed to divide once. After this one division, the DNA collected was all hybrid DNA, that is, it will contain both 14N and 15N. And if you picture it, one DNA molecule has 2 strands, therefore one strand would be from 14N and the other strand would be from 15N. That is what hybrid means. So the first generation is equal to 100% hybrid DNA. Then in a second experiment, they did the same thing but allowed it to divide 3 times. And they are asking what is the composition of this DNA after the third division. So if we look at the options, A obviously cannot occur, which I will explain to you later onwards in this question. B represents second generation. C is for the third generation, and D is more than 3 generations. And why is that? Let's look at the given diagram over here. Initially it would be all 15N, since it is grown in a medium containing nitrogen 15, and over here my key is that 15N is represented by red, 14N is represented by blue. So initially it is all red. One DNA molecule will consist of 2 red strands. Then after it is placed in 14N, which is represented by blue, the first generation will consist of 100% hybrid DNA, as I mentioned earlier. That is, it will have both 14N and 15N. One strand is from red, one strand is from blue. So the first generation is 100% hybrid. Second generation, as you can see over here, it will be 50-50. 50% being hybrid and 50% being blue DNA molecules. And blue DNA molecules means 14N. Therefore, that is the second generation. And if you go to this given diagram over here, it is best represented by diagram B. 14N being 50%, 14N and 15N, that is hybrid, being 50%. So second generation as mentioned. And third generation, it will be 75% being 14N. As you can see over here, there are lots of blue DNA molecules and there's only 2 hybrid, and that is around 25% if you carry out the math, because overall there are 8 DNA molecules, 2 being hybrid and the rest 6 being light or 14N. Therefore, if you convert it into percentages, you would get 75% being 14N and 25% being hybrid, that is both red and blue. And that is for the third generation. Since the question refers to what composition of DNA is obtained after multiplying by 3 times, it is asking for the third generation. Hence the answer is C. D will be more than 3 generations because you can obviously reason it out. If in C it is 75% of 14N and over D it is close to around 100%, that means it has been done more than 3 times, because if you allow it to keep on dividing and dividing, the generations after the third generation will have more of the light DNA molecules than the hybrid.
And as the question is asking about the third generation, the answer is C.
Why cannot it be A again? Because A obviously cannot occur if you go to the diagram given below. As in A, it shows the hybrid being higher than the light, which cannot occur at all, because after it is 50-50 at the second generation, the hybrid keeps on decreasing in percentage. It doesn't increase. Therefore, A cannot obviously occur. Question 3. Which statement about nitrogenous bases is correct? A. Adenine is a pyrimidine with a double ring structure. B. Cytosine is a purine with a double ring structure. C. Guanine is a purine with a single ring structure. D. Uracil is a pyrimidine with a single ring structure. Over here again you have to remember Gapur, which means that adenine and guanine are purines, and all purines have a double ring structure, whereas pyrimidines have a single ring structure. Therefore, adenine is a pyrimidine is wrong because adenine is a purine. Option B, cytosine is a purine, that is also incorrect because it is only guanine and adenine that are purines. Option C, guanine is a purine. Yes, that is correct. With a single ring structure, that is incorrect because all purines have a double ring structure. And we are left with the only possible answer being D. Uracil is a pyrimidine. Yes, that is correct. With a single ring structure, that is also correct because all pyrimidines have a single ring structure. Hence the answer is D. Question 4. Rifampicin is an antibiotic used to treat tuberculosis. It works by inhibiting RNA polymerase in bacteria. Which of these processes will be directly inhibited by this antibiotic? 1. ATP synthesis. 2. Transcription. 3. Translation. RNA polymerase is an enzyme which is only involved in the process of transcription during protein synthesis. Hence, the process directly inhibited would be transcription, and hence the answer being C. 2 only. Question 5. Michelson and Stahl investigated DNA in bacteria. They grew bacteria in a medium with only heavy nitrogen 15N until all of the bacterial DNA contained only heavy nitrogen. These bacteria were then moved from the heavy nitrogen medium and cultured in a medium with only light nitrogen 14N. Some bacteria were collected from each of the next 3 generations and their DNA was analyzed. Hybrid DNA contains both heavy and light nitrogen. Which row shows the correct DNA of the first and third generations? So over here you have to refer to the previous question for the explanation, that is by referring to that diagram which I have drawn in one of the earlier questions. So the DNA of first generation, if you recall, as we mentioned in the previous question, first generation will always be all hybrid, that is 100% of hybrid. Therefore the answer being either A or B. And DNA of third generation was also asked in the previous question, and as we found out over there, the DNA of the third generation will be 3/4 light, that is 75%, 1/4 hybrid, that is 25%. Hence the answer is B. Do refer to the above diagram if you want to go to this again. Question 6. Different tissues in a plant were supplied with a radioactively labeled substance to identify which tissues were actively synthesizing mRNA. Which radioactively labeled substances would be most suitable for this experiment? 1. Adenine, 2. Ribose, 3. Inorganic phosphate, 4. Uracil. Question refers to which is specific to mRNA. Hence, it cannot be adenine as DNA also has the base adenine. And it cannot be inorganic phosphate because even DNA has phosphate groups. Since the question is specifically targeting the molecule mRNA, it should be the one specific to RNA and not DNA. Therefore, the only one specific to mRNA would be uracil, as only RNA has the base uracil instead of thymine, and only RNA has a sugar being ribose, whereas in DNA it is deoxyribose. Therefore, the radioactively labeled substances which would be most suitable for this experiment are ribose and uracil, that is 2 and 4 only, leaving us with the answer being C. Again, why is that? Because the question is referring to radioactively labeled substances which help to identify actively synthesizing mRNA. And there is a difference between mRNA and DNA. DNA is made out of a sugar known as deoxyribose, RNA is made out of a sugar known as ribose, and their bases would be slightly different. RNA has the base uracil instead of thymine, whereas DNA has the base thymine instead of uracil, and both do have the base adenine and both do have phosphate groups. Therefore, adenine and phosphate groups cannot be specific and suitable for this experiment because it would help to identify both mRNA and DNA. However, the question is targeting only mRNA. Therefore, the answer is ribose and uracil. Question 7. Electron micrographs may show large numbers of ribosomes forming chains along mRNA molecules. What is the advantage of this arrangement compared to when ribosomes appear singly on the mRNA? The only advantage is that the process of translation is carried out quicker, because the more the number of ribosomes present to act on the mRNA strand would be quicker translation, which means that polypeptides can be produced more rapidly. Therefore, if you go to the answers A, B, C and D, the best possible answer that describes this is answer D. Polypeptides can be produced more rapidly. The advantage of this arrangement of having many ribosomes that act on a given mRNA strand is faster translation process. Faster translation means the polypeptides are produced more rapidly. Therefore, the answer being D. Question 8. Which row is correct for adenine? The question refers to adenine, and if we recall Gapur, adenine is a purine and adenine binds with T or U using double bonds only. Therefore, does it have a single ring structure? Well, do purines have a single ring structure? That is no. Purines have a double ring structure. Therefore, it should be incorrect under has a single ring structure. Is adenine a purine? Yes, that is correct. And does adenine bind to its complementary base with 3 hydrogen bonds? That is incorrect, because adenine binds to its complementary base using double bonds or 2 hydrogen bonds. Therefore, the answer being C. Has a single ring structure? Incorrect. Is a purine correct? Joins to its complementary base with 3 hydrogen bonds? Incorrect. Question 9. What is the maximum number of hydrogen bonds in a length of DNA containing 700 nucleotides? The keyword over here is maximum number and 700 nucleotides. Although quite tricky, you should break down the question into its key parts. The question mentions 700 nucleotides, however hydrogen bonds form between 2 nucleotides or between a pair of nucleotides. So we should find out the nucleotide pairs. How do you find out the amount of nucleotide pairs? Well, you just divide the given number of nucleotides by 2, because a pair consists of 2 given items. So 700 divided by 2 will equal the nucleotide pairs, and that is equal to 350 pairs. If we assume all the bases to be C and G, since they are asking for the maximum, then only we get 3 hydrogen bonds between a pair, because between the bases it can only be either a double bond, that is 2 hydrogen bonds, or a triple bond, that is 3 hydrogen bonds. And the question is asking for the maximum. So we have to imagine that all the bases would be cytosine and guanine, because cytosine binds to its complementary base guanine using 3 hydrogen bonds. Therefore, 350 pairs multiplied by 3 hydrogen bonds will give you 1050 hydrogen bonds being the maximum. Hence the answer is C, 1050 hydrogen bonds in a length of DNA containing 700 nucleotides. Again, in these kind of questions, you should check out if the number given is of nucleotides or nucleotide pairs. If it is nucleotides, you should convert it into nucleotide pairs by dividing it by 2, and then you should multiply it by either 2 or 3. 2 being for minimum number of hydrogen bonds, 3 being for maximum number of hydrogen bonds. Question 10. Which statements concerning DNA and RNA are correct? 1. Adenine and guanine are bases that have a double ring structure. Cytosine, thymine and uracil are bases with a single ring structure. 2. An adenine nucleotide from DNA is the same as an adenine nucleotide from RNA. DNA adenine pairs with thymine and RNA adenine pairs with uracil. 3. The base pairing that occurs in a double DNA helix and when RNA is synthesized during transcription is always according to the rule that a purine pairs with a pyrimidine. 4. The two polynucleotides on a DNA molecule run in opposite directions so that the double helix formed has 2 strands that are parallel to each other. This question requires careful reading and analyzing of the given statements. The bases of DNA and the bases of RNA are not the same as they differ in size. Therefore, 2 is incorrect because 2 mentions an adenine nucleotide from DNA being the same as an adenine nucleotide from RNA, which cannot be true because the bases of DNA and bases of RNA are not the same as they differ in size. Therefore, answers having 2 in it are incorrect, leaving us with the only possible answer being C, 1, 3 and 4. Adenine and guanine are bases that have a double ring structure. That is correct because adenine and guanine are purines. If you recall Gapur, you will get that adenine and guanine are purines, hence double ring structure. Cytosine, thymine and uracil are bases with a single ring structure. That is correct. Let's look at 3. The base pairing that occurs in a double helix and when RNA is synthesized is always according to the rule that a purine pairs with a pyrimidine. That is correct because if you recall the complementary base binding rule, A binds with T or A binds with U and C binds with G. So again, it does follow the rule that purine pairs with a pyrimidine. And if you look at option 4, the two polynucleotides on a DNA molecule run in opposite directions so that the double helix formed has 2 strands that are parallel to each other. Yes, and this is known as antiparallel because they are parallel but they run in opposite directions. Hence they are known as antiparallel, and that is a correct statement. Hence the answer is C, 1, 3 and 4 are correct. Question 11. 2 sets of bacteria were grown using different types of nitrogen containing growth media. One set was grown in a medium containing the heavy isotope of nitrogen 15N until all the DNA was labeled. The other set was grown in a medium containing the light isotope of nitrogen, that is 14N, until all the DNA was labeled. The DNA from each set of bacteria was extracted and centrifuged. The diagram shows the position in the centrifuge tubes of this DNA. So as you can see over here, the diagrams are given. DNA with 14N being at the top and DNA with 15N being somewhere at the bottom. Bacteria with 15N labeled DNA were transferred to a medium containing 14N and allowed to reproduce once. The DNA of the new generation of bacteria was extracted and centrifuged. Which tube shows the position of DNA from this new generation of bacteria? So although quite a big question, the main key part of this question lies towards the end. Was allowed to reproduce once. What is the position of DNA from this new generation of bacteria? So if you recall in the previous questions, after dividing once, what happens? You get 100% of hybrid DNA. First generation is equal to 100% hybrid. So it is only one position that is possible, which is option D, because all the others do not represent the DNA being hybrid. Because if it is hybrid, it will consist of both 14N and 15N strands. That means it will be somewhat towards the middle. Because if you combine 14N, that is at the top of the tube, with 15N, that is at the bottom of the tube, you would get a position being somewhere in the middle, because it is hybrid and it is 100% of hybrid. whereas A and C shows 2 positions, which is not possible because it should be 100% hybrid, therefore only one position is possible. B shows the same position as the DNA with 14N, which cannot be true, because if it is hybrid it will have both 14N and 15N. So if you combine 14N with 15N, you get a position being in the middle, and that is only if it is 100% hybrid. Hence the answer is D. Question 12. The diagram shows a nucleotide sequence of a small section of a gene which is transcribed. The table shows the amino acids coded for by 10 mRNA codons. What is the order of the 4 amino acids in the polypeptide translated from this small section of a gene? So the given diagram is of a mRNA strand. Then from left to right you have to separate it into 3 bases to form one codon, because one codon is equal to 3 bases. Remember that carefully. So from left to right, as you can see over here, I have split it into its codons, that is 3 bases. So GCG is one codon, followed by CGC, followed by GGC, then followed by GCG. And these given codons, you have to match it with the table over here, because the table also shows the mRNA codons, and what we have obtained over here is also mRNA codons. Therefore, after you carry out the matching process, you would get the amino acids as being ARG, followed by ALA, followed by PRO, followed by ARG. Hence the answer being C. Again, this question is pretty easy. Separate it into 3 bases to form one codon. Then you should match the given codon with the table codons to get the corresponding amino acids. And if you carry out the matching process correctly, you would get the answer being C. Question 13. Which statement explains why DNA replication is described as semi-conservative? Semi-conservative replication. The meaning of it is that each new DNA molecule formed has one parental strand, that is from the original molecule, and the other strand is a newly synthesized strand. Again, semi-conservative replication means the new DNA molecule formed will have one strand from the parent DNA molecule and the other strand being a newly synthesized strand. Therefore, the best possible answer being answer D. The template for each new molecule of DNA is one strand of the original molecule. Why are the other answers incorrect? Half of each original strand is conserved in each new molecule of DNA. It's not half of each original strand that is conserved. A DNA molecule has 2 strands, therefore one strand will always be conserved. Therefore A is incorrect. B. Half of the base sequence of each strand is conserved in each new molecule. That is also incorrect, because semi-conservative replication means that one parental strand is conserved while the other is newly synthesized. C. Only one strand of DNA is used as a template during replication. That is incorrect. During DNA replication, both strands are used as templates. That is why after a DNA molecule replicates, you would get 2 DNA molecules, not 1. If one DNA molecule replicates, you get 2 DNA molecules. And answer D, the template for each molecule of DNA is one strand of the original molecule. That is correct, because if you recall the process of DNA replication, the DNA molecule, it unwinds and unzips, forming 2 strands. Both strands would act as templates, and since 2 DNA molecules are formed, only one strand from the original molecule would be conserved, because from the given 2 strands, one would go to a new DNA molecule, the other would go to a other DNA molecule. And for further visualization and for diagrams, do refer to your textbook. It should probably have the DNA replication diagram, which will help you understand this question more clearly. Nonetheless, the statement that explains why DNA replication is described as semi-conservative is answer D. Therefore, the correct choice being the template for each new molecule of DNA is one strand of the original molecule. Question 14. Which statements about tRNA are correct? 1. Hydrogen bonds between bases temporarily hold tRNA against mRNA. 2. The base sequences in the tRNA molecules are the same as the base sequences in the mRNA that is being translated. 3. The specificity of the tRNA molecule for glycine and the specificity of the enzyme that loads glycine are both necessary for correct loading. 2 is obviously incorrect, because the bases on tRNA are following the complementary base pairing rule. Hence, it cannot be the same. because 2 mentions the bases on tRNA and the bases on mRNA being the same when translation process is carried out. However, when translation is carried out, complementary binding occurs. mRNA has codons and tRNA has anticodons. And why is it called anticodons? Because it doesn't have the same bases as that of the codons. Because during the translation process, it carries out this binding rule: A binding with T or U and C binding with G. Therefore, the bases cannot be the same and it's impossible to be the same. Therefore, statement 2 is incorrect, leaving us with the only possible answer being C, 1 and 3 only. Hydrogen bonds between bases temporarily hold tRNA against mRNA. Yes, that is correct. The specificity of the tRNA molecule for glycine and the specificity of the enzyme that loads glycine are both necessary for correct loading. Yes, that is also correct. And as you can see over here, we have no other choice but to go for answer C, because answer 2 is obviously incorrect. It is impossible to have the same bases in both the tRNA and mRNA when it is being translated, because when translated it will follow the complementary base pairing rule. And what is that rule? A binds with T or U, C binds with G. Therefore, the answer is C, 1 and 3 only. Question 15. What is the maximum number of hydrogen bonds in the length of DNA containing 700 base pairs? Over here, this question is different from the previous one, because the previous one mentioned about just the nucleotide number. However, over here it refers to the base pairs. So you just multiply it by 3 because the question refers to maximum. And if you recall earlier, if it is maximum, you should multiply by 3 hydrogen bonds. If it is minimum, you multiply by 2 hydrogen bonds. And since the base pairs are already given, you don't have to do any division by 2 or anything. Just multiply it straight away by 3 and you would get 2100 hydrogen bonds. Therefore, the maximum number of hydrogen bonds in the length of DNA containing 700 base pairs is equal to 2100 hydrogen bonds. The answer being D. Question 16. What is the common component of the 3 molecules RNA, DNA and ATP? The common molecule in all 3 of them is phosphate, because all have phosphate groups. Whereas if you look at adenosine is only present in ATP, whereas in RNA and DNA it is called adenine, not adenosine. Adenosine is where adenine binds to the sugar unit, which is only specifically present in ATP. Therefore, it is not common in all 3. B. Hydrogen bonds. Hydrogen bonds are not present in RNA, therefore it is not a common component in all 3 of the molecules. Phosphate, as I mentioned, is present in all 3. Remember that carefully. Phosphate is present in RNA, phosphate is present in DNA, and phosphate is present in ATP by the mention of its name, adenosine triphosphate. ATP stands for adenosine triphosphate. That means phosphate will obviously be present in that too. D. Ribose. Ribose is only present in RNA and ATP. In DNA, it is of a sugar known as deoxyribose. Therefore, ribose is not present in DNA. Hence, it cannot be a common component. Therefore, the answer is C. Question 17. A length of DNA was analyzed and sequenced. 24% of its nitrogenous bases were adenine. Which percentage of the bases will be cytosine? Remember the complementary base pairing rule, which is A binds with T, C binds with G. Therefore, if there is 24% of adenine, then there is 24% of thymine. Hence, the remaining percentage would be 100% minus 48%, leaving us with 52% for both cytosine and guanine. Hence, for cytosine alone, you would have to divide it by 2. 52% divided by 2 is equal to 26%. Hence, the percentage of bases that will be cytosine is 26%. The answer being B. Again, since A binds with T, 24% of adenine means 24% of thymine, because there is a complementary base pairing rule. So A and T altogether would equal 48%, because 24 of each. Then the remaining would be allocated to C and G, because there are no other bases in DNA. The question refers to DNA. So DNA cannot have uracil. Uracil is only present in RNA. Therefore, in DNA, the 4 bases are adenine, thymine, cytosine and guanine. And we have already got adenine and thymine being 48%. Therefore, the remaining would be for cytosine and guanine. And how do you find out the remaining? Well, 100% minus 48% will give you 52% for both cytosine and guanine. However, the question refers to the percentage of bases that will be cytosine only, not both cytosine and guanine. Therefore, in order to find for cytosine alone, you have to divide the remaining 52% by 2, leaving us with 26% being for cytosine and 26% for guanine. Therefore, the answer is B, 26% for cytosine. Question 18. The RNA triplet UAG acts as a stop codon terminating the synthesis of a polypeptide. The diagram shows a strand of DNA which codes for 4 amino acids. Where would an insertion mutation of a thymine nucleotide result in the termination of translation? Given is a DNA strand as mentioned in the question, because the question says the diagram shows a strand of DNA. So you have to carry out the complementary base pairing rule and find out the mRNA strand, which would be AGG, UGA, GCU, ACG. That is, after transcription of a DNA strand, the RNA strand should follow the complementary base pairing rule, which I have found over here. Then, insertion of a thymine nucleotide would result in the corresponding base in RNA strand being adenine, because the complementary base of thymine would be adenine. Therefore, wherever thymine is introduced, the corresponding base in this RNA strand would be adenine, and you should try inserting thymine into each of these positions A, B, C and D. So if you insert it at A, it would be AAG instead of AGG, because the complementary base of T is A, the complementary base of T introduced again would be A, the complementary base of C would be G. So the first codon would be AAG, not AGG. However, that doesn't result in a stop codon, because the stop codon is UAG. Let's try B. If we introduce it at B, it would be UAG. Yes, that is correct. Therefore, the answer should be B, because the complementary base of A is already U. If you insert it in the middle of A and C, you would get the complementary base being A again. So UA, and the complementary base of C is G. Therefore, you get UAG if you picture it in your head. Therefore, the answer is B. For this question, draw towards the sides the corresponding RNA strands at each of the positions A, B, C and D, and then group them into codons. That is, one codon is equal to 3 bases. And wherever you get the RNA triplet as UAG acting as a stop codon, that will result in the termination of translation, because stop codon means it stops the translation process. Therefore, the answer is B. Question 19. What is correct for uracil? These kind of questions appear in almost most of the papers. However, the only part of the question that changes is the base. They either ask adenine, or they either ask thymine, or they either ask cytosine or guanine or uracil. Uracil is not a purine but a pyrimidine, because if you recall Gapur, which is that adenine and guanine are purines, uracil is not a purine because it is not part of the word Gapur. The only purines are guanine and adenine. Thus, uracil is not a purine but a pyrimidine. Therefore, under the column it is a purine, it should be not correct, leaving us with the answer being either B or D. And pyrimidines have a single ring. Therefore, under the column it has a single ring structure, it should be correct, leaving us with the only possible answer being B. Has a single ring structure? Correct, because it is a pyrimidine. Is it a purine? That is not correct, because it is a pyrimidine again. It joins its complementary base with 2 hydrogen bonds. That is also correct, because uracil binds with adenine using 2 hydrogen bonds or using double bonds. Therefore, the answer is B. Question 20. The table shows the tRNA anticodons for 4 amino acids. A cell makes a polypeptide containing the amino acid sequence shown: asparagine, threonine, proline, and finally glutamic acid. Which sequence of bases on the transcribed strand of a DNA molecule could code for this part of the polypeptide? Given is a DNA strand, as T is only present in DNA, not RNA. So all A, B, C and D are DNA strands. The table shows tRNA anticodons. So first you have to translate the DNA molecule given over here into tRNA, which is simple because the bases are the same in both DNA and tRNA, except that T is replaced with U because T is not present in RNA at all, whereas in RNA it is replaced by U. So wherever there is T present in this given DNA molecule, you convert it into U and then you group it into 3 bases, because a codon or an anticodon is equal to 3 bases, and then you check whether you get the following amino acid sequence. For example, the first amino acid, that is asparagine, starts with UU in its tRNA anticodons. If you look at the table, tRNA anticodons for amino acid asparagine is UU in both the anticodons, hence it should be TT for the DNA strand, therefore either being C or D. Next comes threonine. Threonine also starts with U in all of its tRNA anticodons, therefore it should be T for the DNA strand. Therefore, following TTA, you should get again T, which is only possible by the answer D. Therefore, the answer is D. C mentions TTA, but it is followed by the base C, which is not correct. It should be the base T. Therefore, C is incorrect, leaving us with the answer being D. Question 21. Which row shows the correct combination? Triplet code, codon, anticodon. The terminologies are: triplet code is only for a DNA molecule, codon is for a mRNA strand, anticodon is for a tRNA strand. Therefore, the best possible answer being A. Triplet code DNA, codon mRNA, anticodon tRNA. Question 22. What does the enzyme RNA polymerase synthesize? RNA polymerase is only present in the process of transcription, and transcription means making mRNA from a DNA template. Therefore, if you go to the answers A, B, C and D, that answer is shown by answer C, mRNA from a DNA template. The other answers A, B, D are incorrect because it does not represent the correct function of the enzyme RNA polymerase. RNA polymerase synthesizes a mRNA strand. And how does it synthesize a mRNA strand? It is from a DNA template, because the whole process of transcription occurs in the nucleus, and in the nucleus there is a DNA template, and then the product of transcription is mRNA, and that process is aided by the enzyme RNA polymerase. Hence, the answer is RNA polymerase synthesizes mRNA from a DNA template, that is C. Question 23. The diagram shows part of a DNA molecule. How many hydrogen bonds are involved in holding these strands of DNA together? Over here you have to apply the complementary base pairing rule, where A binds with T using double bonds, C binds with G using triple bonds. Remember that carefully. A binds with T using double bonds, that is 2 hydrogen bonds. C binds with G using triple bonds or 3 hydrogen bonds. And how many hydrogen bonds are involved in holding these 2 strands together? Well, let's carry out the math. A binding with T using 2 hydrogen bonds, G binding with C using 3, then C binding with G using 3 again, C binding with G again using 3 hydrogen bonds. Therefore, 2 plus 3 plus 3 plus 3 is equal to 11 hydrogen bonds. Therefore, the answer is A. Question 24. Diagram shows part of a DNA molecule. Which row correctly identifies the structures labeled 1, 2, 3 and 4? A base, a sugar and a phosphate all together make up a nucleotide. That is more of a side information so that you remember what a nucleotide is made up of. However, the main part of the question is, what is 1, 2, 3 and 4 representing? 1 represents pentose sugar, hence it is deoxyribose, because the diagram is part of a DNA molecule, not RNA. If it was RNA, it would be ribose sugar, but for DNA it is deoxyribose. Hence 1 being deoxyribose sugar, leaving us with either B or C. 2 is obviously a phosphate group. 3 is cytosine, not adenine, because as you can see, after they have shown 3, they have drawn 3 dotted lines. That means 3 bonds are shown, which can only belong to either C or G, because C binds with G using 3 hydrogen bonds. and A binds with T using 2 hydrogen bonds. Therefore 3 must be either C or G, and since answer B mentions adenine, for 3 that is incorrect. It can only be answer C, cytosine for 3, and for 4 it is thymine, because you can see 2 hydrogen bonds or 2 dotted lines. Therefore the answer is C. 1 being deoxyribose sugar, 2 being phosphate, 3 being cytosine because there are 3 hydrogen bonds shown next to 3, 4 being thymine because there are 2 hydrogen bonds shown next to 4. Question 25. Which statements about tRNA structure are correct? 1. There is a binding site for the attachment of a specific amino acid as well as a different binding site for the attachment to the ribosome in order to allow translation to occur. Yes, that is correct. There is a ribose phosphate backbone with strong covalent phosphodiester bonds and areas within the polynucleotide chain where base pairing by hydrogen bonding occurs. No base pairing by hydrogen bonds occur within a tRNA molecule. Therefore 2 is incorrect, because question refers to within a tRNA molecule or about tRNA structure, and inside a tRNA there is no base pairing by hydrogen bonds. Therefore 2 is incorrect. 3. There is a section known as an anticodon. Yes, because anticodon belongs to tRNA. If you recall an earlier question we discussed about the codon, anticodon and the base triplet. Therefore yes, in tRNA it is known as anticodon, that contains the same triplet of bases as the triplet of DNA bases that has been transcribed to produce the mRNA codon. Yes, that is also correct, because same bases as that of DNA bases, with the exception that T is replaced by U in tRNA. Therefore, since answers B, C and D have statement 2 in it, they are all incorrect, leaving us with the answer being A, 1 only. Question 26. Which is not a description of a gene? A length of DNA which carries coded information as a sequence of nucleotides that can result in the formation of a polypeptide chain. B. Any section of a molecule that has 2 strands, each with a sequence of nucleotides that are complementary to each other and are held together by hydrogen bonding. C. A sequence of nucleotides which can be copied by complementary base pairing and then be translated at a ribosome. D. A sequence of nucleotides that can be transcribed using a polymerase enzyme and free activated nucleotides, and which results in the formation of a messenger RNA molecule. Over here, answer B mentions basically a section of a DNA molecule which doesn't really describe a gene, and the meaning is not complete, whereas the others do describe the gene. Because answer B talks about a molecule that has 2 strands with a sequence of nucleotides that are complementary to each other and are held together by hydrogen bonding. That is basically the meaning of a DNA molecule, because in a DNA molecule also 2 strands are present with a sequence of nucleotides complementary to each other held together by hydrogen bonding. And since the question refers to which is not a description of a gene, the answer is B. A, C and D do somewhat describe a gene. A being a length of DNA which carries coded information that can result in the formation of a polypeptide chain. That is correct. C. A sequence of nucleotides which can be copied by complementary base pairing and then be translated at a ribosome. Yes, that is also somewhat correct. D. A sequence of nucleotides that can be transcribed using a polymerase enzyme, which is known as RNA polymerase, and free activated nucleotides, which results in the formation of mRNA molecule. That is also correct. So the answer which is not a description of a gene is answer B. Question 27. Which row in the table correctly shows situations in which both DNA and RNA are both involved? Columns given are replication, transcription and translation. DNA is only involved in the process of transcription and replication, whereas RNA is only involved in transcription and translation, not replication. Since the question refers to both DNA and RNA being involved, it would be only in transcription. Therefore, the answer is C. Replication not involved, transcription involved, translation not involved. Question refers to both DNA and RNA being involved. And again, DNA is only involved in the process of transcription and replication. RNA is only involved in transcription and translation, not replication. And if you sort of create the Venn diagram over here, the intersection between these two would be only transcription. Therefore, both DNA and RNA are involved in transcription. Hence, the answer is C. Question 28. The following statements describe events that take place during DNA replication and transcription. Which row is not correct? A. Adenine pairs with thymine. B. Both DNA polynucleotide chains act as templates. C. The original DNA molecule is changed after the process. D. Uracil pairs with adenine. The original DNA molecule doesn't change after having completed a process such as replication or transcription. However, if you look at option C, the original DNA molecule is changed after a process. For DNA replication, they have put no, and for transcription, they have put yes. However, the DNA molecule doesn't change at all after completing a process such as replication or transcription. Therefore, the row that is not correct and is obvious over here is row C. Question 29. Which statements about complementary base pairing are correct? 1. Purines and pyrimidines are different sizes. 2. It occurs during translation. 3. The base pairs are of different length. 4. Uracil forms 2 hydrogen bonds with adenine. Remember this carefully. The base pairs which are complementary in nature are of the same length. Therefore, if you go to statement 3, they have mentioned the base pairs are of different length, which is incorrect. Therefore, answers having 3 in it are incorrect, leaving us with the best possible answer being B, 1, 2 and 4 only. Yes, purines and pyrimidines are of different sizes. Complementary base pairing does occur during translation. Uracil forms 2 hydrogen bonds with adenine. That is also correct because A binds with T or U using double bonds or 2 hydrogen bonds. Therefore, again the answer is B, 1, 2 and 4 only. 3 is incorrect because the base pairs are of the same length. They are not different. Question 30. What is the minimum number of base substitutions required to change the nucleotide sequence of the HbA normal allele to the HbS sickle cell allele? Always, if the question mentions about substitution mutation or the minimum number of base substitutions to change from normal allele to sickle cell allele, the minimum is just one base being substituted. Remember that carefully. For substitution mutation, the minimum is one base. Hence, the answer is A. And that is the end of this paper. Hope you found this video useful. If you did, leave a comment down in the comment section below and let me know your thoughts, and also from which country you're watching this video right now. Thank you for watching and stay tuned for more.